speaker is Alex Jourdon, who works with Flora Vaccarino up at Yale. And I think most of us, or many of us on the call, are probably aware of their publication that recently came out using organoids, not only in autism or in cell lines that have been engineered to harbor autism-related mutations, but in particular families where there's an affected son and unaffected fathers. And something else I'm very sure he'll focus on is that there's another layer of order in their samples, is that some of the families have a macrocephalic autistic son and others have a normocephalic autistic son. And it appears that the mechanisms by which things have been knocked off balance might be sort of equal and opposite in their direction, something similar to that. So with no further ado, I will hand off the screen sharing to Alex. Thanks for being here, Alex.
Thank you. Thank you for organizing this. This is a really interesting topic for me and for the lab. And so I will start sharing my screen. Okay. Okay, let me know if you can hear everything correct.
Everything is good, yeah.
Perfect. So hello, everyone. So my name is Alexandre Jourdon. I work in the lab of Flora Vaccarino at Yale. And today we are going to thank you very much for the organizer to start this series on developmental diversity. And today I would like to present a project that we developed in the lab with my colleague Fenan Wu and Jessica Mariani that talked about modeling idiopathic autism in forebrain organoids to see the effect that the disorder can have on developments. And it particularly fits in this topic of developmental diversity, as to carry this project where to differentiate more than 26 individuals, so using many different lines. So as you probably know more than me about autism spectrum disorder, it's like a disorder that is diagnosticated during the early childhood. What's really interesting is that it's a majority of idiopathic cases when you don't really know which genes is actually causing the disease, and there's like a polygenetic risk and many unclear pathogenesis. But there is still light in this disease. As you know, there's like a lot of studies that have been released, notably like large genomic studies, exomic and whole genome sequencing studies. And there's been like more than 1,000 genes that have been associated with the disease with different degrees of confidence. And they are notably grouped, for example, in the spheroid database. And another important aspect of this disease is that it seems that the genes that are affected probably have an effect during the prenatal developments, which then, of course, makes it perfect to study in vitro. And the second aspect is that it seems to be related to an imbalance, or actually an involvement of both excitatory and inhibitory neuron lineage. So in our lab, as in many labs in this talk, we are working with organoids, and particularly forebrain organoids. And as you know, forebrain organoids develop from iPS cells from humans, and it's a 3D models, and it's a system that we've been studying in the lab for many years. And what's important for us and in this series is that it's linking, really the most important thing to remember is that it's always linking human genetics to a system that is pluricellular and developmental in the sense it's constantly evolving systems. And that's why we got interested into developing it. And of course, the technique I will speak mostly about today is single-cell RNA-seq, which is like the best technique to study the complexity of cells that we observe in organoids and to really understand what's going on. So then how do we study autism? In our case, we decided to go for idiopathic cases, and we took a family design approach when we collected iPS cells from a set of families, 13 that were involved in this current study. And each time we compared a male ASD province to its neurotypical father. So this way correcting for part of the variability in the disease and correcting also for genetic backgrounds. There is also a second layer of groupings that we decided to do, is to group ASD based on the head circumference, and notably classifying them as ASD with macrocephaly when they have like above 90th percentile in terms of head circumference, and ASD with normocephalic in contrast. So out of that, we differentiated like a 26 iPS line from each of those individuals, and we formed like forebrain organoids that we collected at 3 time points that we named TD0, which is the beginning of neurogenesis when we start switching to a neurogenic medium. and TD30, so 1 month after, and TD60, 2 months after, to really see what's going on during the early neurogenesis steps. And from each of those stages, we collected single-cell RNA-seq using the 10x sequencing platforms, using the recent technology, and we collected roughly 10,000 cells at each of the samples. We did also a multi-omic approach that we collected also many other different types of assays, including bulk RNA-seq to really is cheaper and allow more replications, whole genome sequencing to characterize the variants in each of those individuals, and also more epigenomic information, collecting a lot of ChIP-seq on multiple marks, and single-cell attack-seq that we start collecting now to get more single-cell level open chromatin regions. So, of course, this is a large project that's been ongoing in the lab for many years, and it's been involving many people. Notably, we have Jessica Mariani and Fenan Wu that did also a lot of the analysis. And the study I will present today has been mostly in group, as Carlo was saying in a recent publication, but it's an ongoing project. It's also been done in collaboration with Alexei Abizov at the Mayo Clinic, and which analyzed a lot of the whole genome sequencing. So what did we get? So when we characterize, of course, those cell types at the beginning, we integrated everything into a single reference that you see here in the UMAP. And we collected like more than 600,000 cells using roughly 72 different libraries. And of course, like every steps in the single cell analysis, we started by characterizing the neuronal diversity, showing that it was reproducing gene expression as we expected, presenting different neural progenitors that evolves along what we can be organized as pseudotime. And of course, we characterize a certain degree of neuronal diversity in our system, showing notably like the presence of excitatory neurons characteristic of the cortical plate, notably expressing, for example, neuroD6, but also excitatory neurons that are more immature and express, for example, relians, such as chiral cells that we characterize as excitatory neurons of the preplates. And finally, we have also a cluster that is expressing inhibitory neuron lineage. And we validated several of those cell types using, of course, immunostaining to show how they were organized inside organoids. And just to let you know, like as I guess Carlo will speak, is that we've been able to upload this data set in Nemo so that you can explore the gene expression in this data set. So of course, to validate at the time the characterization of a cluster, we also correlated this expression level of each cluster with what was available in terms of fetal brains. And that was notably 2 data sets from the Kriegstein labs that allow to calculate the correspondence between the cluster identified in organoids with the cluster identified in the brain. And now it's really interesting also for the field. Something that's really important is like over the last months, there have been so many data sets that have been released of fetal developments and like gross diversity of regions collected. That is really interesting to further annotate our cell types. So then we enter in really the core of like what's interesting us today is like the source of variations in organisms, and notably once we have characterized all those cell types, how do they vary actually between samples? And that's what's really an interesting thing to see is that while we differentiated all those lines under the same protocol, which involve like a dual SMAD inhibition, trying to target as much as possible toward the anterior portion of the forebrains, we actually saw a gross variability in terms of cell composition, as you can see here on the left, which each samples is one row and it's colored by the cell compositions. So of course you have like the first distinction that you can see that's driving, of course, the organization of cell composition is time, as of course at early stage organisms are mostly composed of progenitor cells, and as they differentiate you get more and more neuronal cell types. And what's interesting is that then when you take a later stage is that organoids are spread, of course, by the proportion of neural cell types or the different subtypes that we identified. And you can see here on this PCA plot how they're organized. And one interesting thing to see is that what was driving this polarization between different samples was the presence of, for example, inhibitory neurons, excitatory neurons, or other subtypes of cells. And notably, like, for example, cells that have medialization phenotypes. And that's something we can see in terms of distributions. And suggesting that, of course, the organisms, while they are submitted to the same molecules, tend to actually line to line have a tendency to differentiate into different paths, maybe more medialization or more ventralizations in the fate of cells. And of course, we verified that using a bulk RNA-seq, when we did more replication, we verified that there was a more transcriptomic similarity at the bulk level, which correlate highly with cell compositions between samples from the same individual differentiated multiple times compared to different samples. One also things that impact of this cell distribution we noted was that family, so the genetic background in parts, drives more similarity to each other. So the two individuals from the same family tend to have a more similar compositions on average. So then one thing we thought to exploit in front of this diversity of cellular composition is like what's driving this composition, what's the rules behind it, and how can that inform how the brain builds itself. And so really at that time, it's like we decided to do a really simple approach when we took basically expression in progenitor cells, so in radioglia, and we correlated that with the actual amount of proportion of cells that you observe in the organoids in the same samples. And that's really tells you, for example, if you plot each sample, here is dots, and you plot them based on their proportion of inhibitory neurons and their proportion of excitatory neurons, and then you compare that with the expression of like famous transcription factors in radioglia, you can see here, like in yellow, they being the strongest expressions, that samples that have a high proportion of excitatory neurons, of course, expressed a lot of EMX1 in their radioglia, whereas samples, for example, that expressed a lot of GSX2 will have a large concentration of inhibitory cells. And what's interesting is that, of course, you have cases more complex when, for example, FOXJ1, which is the marker, the anterior markers, will drive both lineage. And so we kind of propagated this analysis to include many more genes and identify in total each genes that are related to each lineage that we observe in organoids. And that's what we term neuron predictor genes. And was notably, this analysis was notably validated by the fact that, of course, you find back some famous transcription factor linked to each of those lineage, including, for example, NeuroD6 that drives excitatory neuron productions, or, for example, GSX2 or DLX and 6-3 that drives inhibitory neurons proportions. So then we attacked the other side that was interesting us. It was like, how do we identify what's the ASD phenotypes? And one thing I showed is that since there is no actually strong divergence between the ASD sample and control samples, we decided to, of course, use our family designs to investigate what was the differential expressions between ASD and controls. And so one thing to really remember in this study is that we try to differentiate as much as possible the neurotypical father with the ASD individuals and done almost everything in parallels. So then we decided to do, of course, a pair differential gene expressions between each individuals that carries ASD with the father. So we repeated this analysis for each of the family that we differentiated, and at the end steps, try to identify the convergence between families to really identify what was the phenotype in commons. And so then we identified these macro ASD phenotypes, and we repeated this analysis for the nomocephalic, and then we decided to compare, of course, the two phenotypes to understand how convergent they were. So the main results is that what we identify in terms of gene expression is like a large increase in macrocephalic ASD individuals in excitatory neurons and cortical plate gene expressions, as you can see here from the increase across all cell types in genes related to those functions. And that was also in contrast to, of course, a large decrease in inhibitory neurons and other subtypes of cells. What was really interesting to see also is that when you compare that to actually normocephalic ASD, you see almost a reverted, although less proeminent phenotypes, and you see notably a decrease in excitatory neurons neurogenesis signature. And that's something that was associated with the early stage with the modification in proliferation putatively, notably with disruption of gene associated with neurogenesis. And so in total, between the two sets of the differential gene expression that we observed from those two cohorts, we saw that there was really a limited overlaps between the two cohorts, with like really most of the DG being the differential expression genes being associated to one or the other cohorts. And so that really pointed to the fact that there could be really two different phenotypes based on this head circumference characteristic for the ASD individual. So one thing, of course, that was corresponding with this differential expression is that when you look then at the cell types and you compare actually cell compositions between the proband and the controls, you see in macrocephalic a decrease in, for example, excitatory neurons of the preplates and an increase in excitatory neurons of the cortical plates, something that is reverted, of course, in the progenitor of normocephalic, in cells of normocephalic individuals. And of course, you can link this change in cell compositions with the gene expression. And for example, if you take intermediate cells, so the intermediate progenitors, you can see that they already carry this change in trajectory. And most of the macrocephalic DG in that clusters actually already starting expressing cortical plate markers, suggesting there was an increase in neurogenesis towards the cortical plates excitatory neurons, whereas like preplate signatures were actually more increase in normocephalic DGs. And of course, when you take then the progenitor cells, you also see an enrichment in their DG with genes that we characterize as being associated with the production of each neuronal subtypes. And notably in macrocephalic, an increase in excitatory neurons predicting productions, genes inside the progenitors in all the different progenitors that were identified. And it's something also that we validate with immunostain, showing that there was an increase in protein associated with neurogenesis of the cortical plates in ASD compared to their controls in the most striking families. So of course, we saw this nice bifurcating of phenotypes, and we were quite interested, but we also tried to look at, is there any way a convergence between all ASD individuals that we identified? And so for this, we merged all our cohort together and tried to identify which genes were in commons between all of them. And what we can find is that this convergence was quite limited. It was only concerning a few dozen genes per cell types. And what was interesting is that maybe the strongest coherent signature that we saw was related to mitotic cell process, cell divisions, and putatively RNA binding functions. So still a little bit generics, but really interesting to see. And it will be something that will be interesting to see as we increase our cohort to see if this is reproduced over many individuals. On the contrary, the genes that were downregulated were on average had a less clear signature in terms of annotations, except maybe for a few genes related to VEGF-A and VEGFR2 signaling pathway. One other interesting feature that we noticed is that while we characterize a broad diversity of differential expressed genes, we saw that there was a certain enrichment for genes that were already associated with ASD from a rare variants whole genome sequencing study that I was mentioning. And this is notably by merging different publications that appear recently to try to identify ASD risk genes and also comparing with the SFARI genes. And what we saw is that there was the strongest enrichments in differential expressed genes from macrocephalic differential expressed genes. And notably, this was occurring stronger in preplate cells, for example, and other subtype than excitatory neurons, which was really interesting to see too. And so this intersection suggests that there is potentially a common path, of course, between idiopathic and rare variance cases. And so that's really interesting to see the degree of convergence. But of course, that drives us to ask an important question is like, what about in our cohorts? Is there any association between what we see at the transcriptomic levels with the mutations that could affect those risk genes? And so we then focus more on the whole genome sequencing aspect. And so we sequence all our IPS lines and called each variants present in each of the whole genome sequencing results and focused on rare variants and notably on genes that were either characterized as syndromics from the literature for ASD or that were associated with DEGs in our results. So the way we compare that is to identify for each individual, of course, we found different variants. And for example, like this is an example of a deletion that appear in a family that affect an important gene, for example, as PTEN. And then what we compare is like, is there any affection of the expression of the genes in the cohorts when you compare to the general distribution that is represented in black. So here each individual in a large dots carry the mutations. And what we saw is that overall, there was no a strong affection of the gene expressions when the individual was carrying a deleterious variance, suggesting that those variants actually do not necessarily have a direct impact on gene expressions and at least do not have an outlier kind of behaviors. It's also, of course, something that we just characterized and just presented to assume that those individuals who are idiopathic is that there is no strong deleterious effect of their mutation on the characteristic that we are studying, suggesting that the rare variants here that we identify are not responsible for the phenotype that we are seeing. And so, of course, that was really interesting to see. And that's where we start also raising back to the complexity of the topic of today is like the diversity of individuals and mutations and how they could impact actually organoids developments. And so it's the only potential cases that was really the strongest was like we identified really large duplications that were affecting that were in the regions of a SFARI genes called POXI. And it was only in one individuals. And what was impressive is that we saw in these large regions that involve many genes, that all the genes of that regions were actually strongly expressed and almost at the highest level of expression across our cohorts, across all cell types. So this thing that in this case, we could really associate that there was potentially a driving force in the transcriptomic phenotypes. And of course, these genes and these regions, all the genes, almost all the genes in that regions were differentially expressed between the proband and the controls that didn't carry these mutations, suggesting that they could have an impact, of course, on gene expression. And so that's a really interesting individuals, notably because POXI is still one of the most robust associated genes with ASD, even in recent publications. And so it will be really interesting to further study this individual. Of course, then when we start going a little bit further and try to understand how the mutations that we identify in each individuals and each genes potentially drive what we see at the level of the cohorts, we have, of course, a difficult step when we have to understand if the genes itself, the mutation itself in the genes will affect gene expressions. And what we see is that in many cases, it's really difficult to find a link, a causative link, of course, at these levels between the mutation that is sequenced and the transcriptomic alteration that we see between, for example, a father that doesn't carry the mutation and the control and the ASD that carry the mutations. Maybe, for example, there are some cases like, for example, you see this duplication in these individuals affecting the gene CHEK2 seems to have created, of course, a large increase in expression compared to the fathers. But of course, this to demonstrate such a thing needs either to have a large cohorts or to be able to study and mutate and reproduce this mutation to really demonstrate this link. And that's where really the difficulty of like linking genetics to transcriptomic in our studies. But overall, what we noticed is that there was not enough of all the differential expressed genes that we identify in our studies. There was not direct explanation at the genetic levels that we could identify easily. And so that suggests that there are other explanations, of course, for the differential expressed genes that we find. Most likely, they are either like downstream effects of other genes mutations or more common mutations that affect a different level, different genes, and that could converge on a differential expression down the line. The other aspect also is that this could be also mutation in non-coding regions. That here, since we focus mostly on coding regions that were higher to predict the effect, it's a little bit more harder to focus on non-coding regions when you have not a really idea of what could be the effect of the mutations. So here I would like to present quickly like another aspect of the study when we try to characterize in brief words the epigenomic informations in the same cohort that we characterized. And that consisted notably to use the large epigenomic information that we collected to try to identify unanswered regions which control gene expressions in the idea of building a regulome where you can link actually an answer to genes and build a gene regulatory network that is based both on coding and non-coding regions. And the idea being that then if you can then replace the DG that we identify in a study in a network, you can really understand which genes are driving the phenotypes that we observed. And so, of course, here I want to present really quickly, like this looks, for example, like this, when you kind of put together both the unanswered informations and the gene expression, and you link them together based on the correlation between, for example, the activity of the unanswered and the activity of the gene expressions. And it's a study that we did at the bulk levels, and we can identify already a large networks of correlations. And so you see, for example, for each gene of interest, you can identify different unanswers, and you can link them to putative upstream transcription factors that are themselves, of course, unanswered identified. So altogether, to bring a large story short, is like we then put together a gene regulatory network using these informations and replace the differential expressed genes that we observe at the bulk levels into a network to try to understand what was the relationship between the different differential expressed genes. And what we identify here in this large network is that when you try then to derive which transcription factor are driving the phenotypes, we of course found back that important transcription factor linked to excitatory neurogenesis were associated with microcephalic ASD. As you can see here, for example, from the strong disruption of BH, LH, E2 and 22, sorry, FOXG1 or EOMS, for instance, that would drive a lot of differential expressed genes downstream in their network. So altogether, to bring the hypothesis, the working hypothesis that we have to right now in the lab, is that compared to neurotypical individual during neurogenesis, we see that ASD with macrocephaly will tend to have potentially an increase in cell divisions that could lead to then an increased production of excitatory neurons of the cortical plates. On the contrary, when you looked at an ASD with a normocephaly, it seems that the opposite could be happening, and that actually there could be maybe a slightly more premature tendency to generate neurons early on of the preplate, for instance, and that would deplete, of course, the progenitor pool and potentially decrease the quantity of excitatory neurons generated later on. Both of them will result in an imbalance in cell type, and that's what we are really interested to look at. And we all would like to validate that in more and more cohorts, as we know that each of these subset might actually be more complex than just a common group. So altogether, the perspective in this work is that we are trying to first exploit even more the unanswered that we characterized to really finish. We have currently a preprint available on unanswered gene regulatory network. And we'd like also, of course, to move to the single cell level, trying to build both from ataxic and single cell RNA-seq cell type level regulatory networks to be really able to understand how upstream genes are driving the differential expression that we are seeing in our cohorts. We're also conscious, of course, that these studies is just a first studies and that we need to replicate it and expand the diversity. We're barely scratching the diversity of human genetics and ASD genetics. And so we are trying to collect more and more ASD individuals. We have currently more than 40 available in our lab and trying to expand that even more. We're also trying to expand with ASD from female individuals and to also extend our differentiation protocol to include other regions or other protocols. Finally, of course, as we are all interested here, is like it's really interesting to see that at some point, with collecting enough information from idiopathic individuals, you can start approaching the ability to classify phenotypes based on organoids readout, and potentially, as I was trying to discuss in the later part, trying to link what you see at the genotype levels with what you see at the phenotypic levels, notably from transcriptions. And of course, in the future, and that's here, I'm speaking long term, the idea of like all this organized work that we're doing in complements with between many labs would be to allow to stratify patients based on, for example, putatively and organoids systems, and of course, go to a specific potentially treatment or at least understanding of each case. On that, I would like to thank you and thank, of course, all the people involved in these studies. That includes in the lab of Flora Vaccarino, like Fenan and Jessica that did a lot of the work on this that printed today. And of course, Alexei in the Mayo Clinic that did most of the whole genome sequencings, and every other people in the lab of Flora. And we also like relied a lot on many of the platform that were Yale and all the families, of course, that were involved and accepted to be part of the study and our funding sponsors. Thank you.
Thank you, Alex. So now we can open it for a few minutes of questions. There's actually one on the chat we could start with. So Aditi from Helena Kilpinen's lab in Finland is asking, I was wondering if you also had head circumference info on the control individuals, and if there was any correlation within the controls between head size and excitatory and inhibitory balance.
So in the end, the macrocephalic individuals often have also a macrocephalic neurotypical father. So macrocephaly is not really a distinctive criteria between the control and the province, and it's more like we classified our individual based on the head circumference of the province. And so we're really interested into that. In terms of the classifying like the relationship between the head circumference and the excitatory neurons neurogenesis in controls, it's really difficult because in this case, like our cohorts become too small to be able to drive these informations. And we're also limited by the way that we cultivate organoids when it's a little bit difficult to evaluate, for example, if there is an impact on organoid size across all our cohorts. And so for that, we need to really, we are kind of like hitting the limits of like human variability in the sense that macrocephalic is really common traits among the population, at least how we defined it. And so we really need more samples to be able to correlate clearly head circumference as a simple phenotypic information with excitatory neuron genesis in the broad diversity of genetic populations of neurotypical populations.
So something you mentioned, not only did you study 26 lines or at least 26 donors here, but you and many others are trying to do more and more of that. Both your own work and some of others on this call look to not only say the production of a particular downstream lineage, but also the initial signaling or organizing events that give rise to those. So this is a question both for you and anyone else on the call. Does this work or your ongoing work indicate to you what kinds of assays you would want to implement if you were screening or assessing a large collection of lines? You know, those are, because some people are trying to get rid of the variability, some people are trying to study that variability, but it's good to know where it is and how to assess it.
Yes, and that's really an important and interesting question. On our side, like for example, we are trying to develop more and more protocol when we're trying to give even more information to the cells during development, trying to restrict them. As I say, for example, in this current cohort, we saw like there was one of the variation, the explanation of the variations was the ability to ventralize or on the contrary, medialize compared to the cortical plate, I'm speaking. And so cells tend to kind of a certain bias. And so what we thought is like, maybe if we kind of start expose them, for example, to sonic hedgehog to try to ventralize them, maybe we can control even more what the line is doing. And the truth is that it's at least from the early results that we saw is that it's actually possible, but it's also the lines still do also what they sometimes want, and they have a certain preference to respond to different morphogenes. So that's really an important question, because we don't have yet the answer how to really control these variations. And we are certainly definitely in our lab at least trying to understand from these variations what are the rules behind, as I showed.
Alex,
I don't know if
Alex,
yeah.
oh, this is Nicola. A really great talk. Thank you. You showed in the single cell RNA sequencing in the UMAP that you could identify progenitor or the resembling the cortical hem, right? You annotated the progenitors like radial glia hem. I was wondering if you also look for markers of the ROSA pattern center. So you show that these cells express markers of the dorsocaudal organizer, but what about the Rosso pattern, the anteroventral organizer? Because these are, you should try to find these markers at T0, right, in the early states, and this maybe can explain at least the variation of the bias, right?
Yes, indeed. We try to look at organizer signature, and here I'm putting like the figure itself, right? When we try to see if there was sign of anterior, posterior, and like different signature than with the knowledge that we had. What we saw is that there was not necessarily a clear organization of our progenitors based on this, and that really probably the strongest signature we saw was really this hem cluster that we really see these medializations, whereas the more subtle, I would say, frontal, for example, organizer or others, we didn't see really a strong signature. At least we could not identify the radial glial cells that was carrying these signatures. And so this was less proeminent in our system.
Thank you.
Thank you. Okay.